Welcome back, my dear students. In this course, let us learn about infertility and assisted reproductive technology. Infertility. So, it is a condition where there is an inability to produce a child or unable to conceive. And the reasons for this could be many, like congenital reasons, physical reasons, and there may be diseases and drugs and immunological reasons also there, and psychological reasons. Because of all these reasons, the people faces the inability or they couldn't able to produce a child. Now let us see what are the defects in males. The first defect is the deficiency in the gonadotrophins. Gonadotrophins are the hormones which regulates the gonads. That means in males it is regulating the testes. And the gonadotrophins are of two types. The first one is FSH, that means it stimulates the gamete production. Whereas the second one is LH, that is luteinizing hormone, and this helps in the production of the hormone by gonads. Okay, so if there is a deficiency in gonadotrophin, the first is the gamete production will be affected, and second one is the hormone production will be affected. And second reason is undescended testes. That means the testicles that has not moved into the bag of the skin, that means scrotum. The testicles has not moved into the scrotum region. So this may be before birth reasons. And next is the defects in the penis. That means the penis disorders. So this penis disorders can also be seen as a birth defect, and sometimes it is because of the skin abnormalities. Next is the blockage of the sperm duct. If the sperm cannot pass through the duct, if the ducts have been blocked, then the sperms cannot pass, so that there will become defect in males. Next is another condition called azoospermia. That means non-motility of the sperms. Sperms will be there, it will be produced, but the major character of the sperm is motility, right? So if the sperms are non-movable, it cannot reach the ovum. Next is oligospermia, that means less sperm production. Next is sexually transmitted diseases. Next, let us see what are the defects which are seen in females. The first is again deficiency of gonadotrophins, that is follicle stimulating hormone and luteinizing hormone. And next is defective vagina. That means if the female is having abnormal vagina, she couldn't able to get conceived. Next is during the prenatal development, these all things are the changes which takes place in the vagina. Sometimes absent vagina is also seen in some cases. Next is defective uterus. That means this is a congenital uterine abnormality. That means which has been found from the fetus itself. That is before birth only these all defects can be seen. Next is the blockage of uterine tube and lack of ovulation. If the ovulation doesn't occur properly, if the good quality ovum is not produced, so that ovum cannot become cannot come in contact with the sperm and cannot get fertilized. Next is the tumors in the uterus. If there are tumors in the uterus, it becomes very difficult to get conceived. Next is the improper development of the uterus. That is the uterus having different shape, structure from the normal. Okay. Next is the sexually transmitted diseases. Then what is the solution for this? That is assisted reproductive technology. If the couples are having these kind of defects, they could be assisted to have their own child through certain special techniques, and these special techniques are known as assisted reproductive technology, ART. So now let us see what are the assisted reproductive technologies which are available to have childless couples to have their own child. The first is in vitro fertilization and embryo transfer, and it is commonly known as IVF-ET technology. That means IVF-ET. See what they are going to do in IVF means. Let me explain you step by step. The first step is the woman is treated with FSH hormone. So this stimulates the ovary to produce multiple ovum, that is called multiple ovulation, and this results in the release of several ova at a time instead of one. Usually one ovum will be produced, right? So if the woman is injected with FSH, multiple ovulation takes place, which results in the release of several ova at a time, and this is also known as superovulation. Next, an incision, a small incision is made near the umbilicus region, and the ova are aspired with the help of an instrument called as laparoscope, and this method is known as laparoscopy. And the ova are collected now, and that collected ova is incubated in a petri dish for few days, and it is incubated in a suitable medium. Meanwhile, the semen is also collected from the donor or from the sperm bank also, and this is kept in a suitable medium again. Then the incubated ova and sperms are mixed in a test tube, and here also the test tube should contain a suitable medium for the fertilization. Then within 24 hours, the ova are fertilized with the sperms in a test tube. This type of external fertilization that takes place outside the body. Here we are conducting this fertilization in the test tube, right? So hence this is known as in vitro fertilization. The fertilized ova or zygote are then transferred into the another suitable medium. In the another suitable medium, they undergo a process called cleavage, that is dividing. Then the cell starts to divide and the fertilized ova reaches 8-celled or 16-cell stage. Then again in out of these, whatever the fertilized zygote are there, no? So out of that about 2 to 3 are selected, and the 2 to 3 healthy fertilized ova are transported to the uterus of the mother. Or else if the mother is having some defects in the uterus, then they will go for the surrogacy, and that mother is known as surrogate mother. Now the ova which is fertilized, which is transferred to the uterus, and which results in the implantation, and implantation again goes for the further development, and this process is known as embryo transfer, because we are transferring the 8 to 16 celled cells to the uterus. Then the embryo which gets implanted develops into fetus, and whatever the others are there, no, they may develop, they may degenerate. So we are just adding 2 to 3 in order to confirm it. Sometimes we can see the triplets, or else we can see the twins. If it is not possible, the other 2 will generate, at least 1 will develop. And this method of producing the baby in a test tube is known as the test tube baby technique. The baby which is produced by conceiving in a culture dish and nursing in the uterus is called as the test tube baby. Now let us go for GIFT, G-I-F-T, that means gamete intrafallopian transfer. So this is also another method of assisted reproductive technology. Here in this method, the sperms and ova are collected and which are introduced into the upper part of the fallopian tube. In the upper part of the fallopian tube, see here, they are collecting the sperms and ovum and they are just transferring to the upper part of the fallopian tube where the fertilization takes place. Here just gametes are getting transferred. Next is ZIFT, Z-I-F-T. Here the zygote intra-fallopian transfer is seen. That means here the fertilization takes place outside. That means eggs are collected and sperms are collected. They are fused in a petri dish. Again they are transferred to the fallopian tube. Okay. After in vitro fertilization, the zygotes are transferred, means that is ZIFT. If the gametes are transferred, means that is GIFT. Next is one more technique called POST, that means peritoneal oocyte and sperm transfer. In peritoneal oocyte and sperm transfer, the peritoneal ovum or oocyte, they are transferred. So this is a fertility treatment method. So it is an alternative to the IVF technique. Okay, here the sperms and eggs are placed into the peritoneal cavity. See here, here the sperms or eggs are taken and fused. Okay, but this fusion takes place in the peritoneal cavity near the fallopian tube, but not in a petri dish. And this is done under the ultrasound guidance. Next one more is DOST, that is direct oocyte and sperm transfer. So here after the retrieval of the egg, that is from the stimulated ovary, and after the retrieval of the sperm, they are transferred directly. This is also under the guidance of the ultrasound. So these are the techniques which are available for the childless couples to have their own baby. Next, one more, the ICSI, that is intra cytoplasmic sperm injection. In this method, this is a specialized procedure. Okay, so in order to form the embryo, they are going to do it in a laboratory. Here the sperms are directly injected to inseminate the female. Okay, and this is done in case of very low sperm counts. Okay, so this can be corrected by using one more technique called as the artificial insemination, AI. See here, in this technique the semen is collected either from a husband or from a healthy donor, and that is artificially introduced either into the vagina or into the uterus, and this is called as intrauterine insemination, IUI. Okay, so these are the options which are available for the childless couples. Though the options are many, the decision by the people in India is limited. Why? Because of some emotional, ethical, religious and social beliefs. So this is about the assisted reproductive technologies. Hope you understood. Thank you for listening.